So, welcome everyone, and thank you for tuning in to the Mid-Atlantic Micro Nano Alliance Virtual Seminar Series. I'm Heidi Leonard, President of MAMNA. Today's seminar will be our fifth of the virtual seminar series. Last week, our presenter Ruben had over 500 views with a lot of questions and a lot of enthusiastic feedback, which was very encouraging to see. And as always, I'm joined here virtually by co-president, Professor Ryan Sokol. So Ryan, who do we have the honor of hosting today?
All right, so hello everyone. Today we have Claire Herr, who is the Claire Booth Luce Assistant Professor in the Department of Mechanical Engineering and Department of Oncology at Johns Hopkins University. She received her BS, MS, and PhD in Mechanical Engineering from UCLA in 2005, 2007, and 2011, respectively. After her doctoral training, she joined the Rollin Institute at Harvard University. Before joining the Johns Hopkins University, she managed the clinical studies funded by the Vortex Biosciences Incorporated company. She's won numerous awards and scholarships, including the Edward K. Rice Outstanding Doctoral Student Award, the 2018 inaugural Johnson & Johnson WeSTEM 2D Scholar Award, the School of Engineering Faculty Award at the 2018 Johns Hopkins Department of Medicine Research Retreat, and the 2019 Hartwell Individual Biomedical Award. So it's my pleasure to introduce you to Claire.
Thank you, Ryan, and thank you everybody for tuning in. My name is Claire, and today I'm going to tell you about inertial microfluidics that my lab are developing to help advancing biomedical research. And before I start, I always have to disclose that I have a financial interest of some of the technology that I'm going to talk about today. So hopefully you guys will understand that my opinion towards this technology is a little biased. And also, I want to thank all the people working behind to make this type of work possible. So my young active group, and then my formal postdocs, and my previous and then current funding sources, and then obviously a lot of collaborator helped us to get where we are. So I always introduce myself as a tool developer because I was trained as a mechanical engineer. And my lab is particularly interested in making microfluidic tools that can help personalize medicine. So we're not treating a patients with these devices, but we're providing building devices so that we can provide right information to clinical researcher doing research so that they can find what's going on with the patient's body and then what will be the best therapeutic option for individual patients. So as an engineer, what I think that my lab can do is finding a right specimen from the patient's body. For example, cells that carrying some kind of pathological or genomic information. So collect those cells that carrying disease information so that we can understand what's going on with the patients. And also we can alter those cells or finding the right things to make those disease can be either ceased or progress very slowly. So today I'm going to tell you about how we purify things from the either bodily fluids, blood or urine. And also afterwards, I'll tell you how we're going to find the right information for individual patients. So first, I'll tell you how inertial microfluids can be used to purify certain target cells. So for this audience, I don't think I need to really explain what microfluidic is, but you might not heard about inertial microfluidics. So the term inertia of that inertial microfluidics comes from the Reynolds number being greater than 1. So Reynolds number is nothing but ratio between inertia force over viscous force from the fluids that's surrounding the object when the object is moving. And inertial microfluidics rise in very small niche of the Reynolds number range. It is still laminar, but it's greater than 1. So the inertia force from the fluids cannot be ignored. So we operate slightly higher range of a lot of other conventional microfluidics are operated at. And what interesting thing happened in inertial microfluidics, if you inject this particulates, the relatively large size compared to the channel dimension. So we usually deal with particles greater than 20% of the channel width. And if we inject them into the thin microchannel at really fast speed. So the typical flow rate or velocity that we're dealing with is a few centimeters per second up to a few meters per second. So when we inject this particle into microfluidic device at fast speed, because of this large velocity gradient that formed across the channel dimension, this particle tend to experience lift force from the fluids. So it's kind of similar to the lift force that airplane flies when they take off. There's a pressure gradient across the airfoil, so they get migrates or lift up into the air. But similar thing happen when the particles are injected into this sharp gradient of the fluid velocity. Then particles start experiencing the lift force that's pushing particle perpendicular direction to their travel direction. And these lift force are in different direction, operates in different directions. So when the particles are near the center, the lift force pushing particle towards the wall is a lot stronger. So a particle gets migrate towards the wall. And then when the particle gets too close to the wall, the viscous uh, layer between particle and channel wall push the particle away from the wall. So since these two forces are acting on opposite direction, particle, as a result, they can find the equilibrium position somewhere these two lift forces are perfectly balanced. And also if the particle is soft, it can change its shape. So they're having additional force making the particle migrates away from the channel wall. So as a result, if you have a particle with all uniform sizes and the mechanical property, they find very distinct distance from the wall and then follow through the channel very uniform speed. But if the particle has different sizes or softnesses, they tend to find slightly different location because their balancing lift force are going to be different. So by using this type of differences of lift force, how it's affecting particle location, we have developed many different devices. And then I'm going to walk you through some of the characteristics of this inertial microfluidics. So first thing that's interesting thing about inertial microfluidics is we can control how many this focusing position we can form by changing channel geometry. So if the channel is perfectly square, then the particle tend to form the 4 focusing positions that's following the symmetry of the particle cross section. But when we reduce aspect ratio, so making the square become a rectangle, then these other 2 particle focusing position previously existing for a square become unstable. So as a result, we end up having only 2 particle focusing positions from the distance away from the channel bottom. This gives us, this is another view. If you're looking at the side way, if the Reynolds number is not perfectly matched, all the particles are flowing at different speed and random location. But if we're inertially focused them, we can form very uniform lattice of the particle flowing with the uniform velocity. And this, by using this phenomena, we can position particles with the identical shape and identical size, or the rigid particle with slightly different shapes, or even the discoid red blood cell, we can focus them in very uniform location and then flow them at identical speed. And as a result, their distance from the wall is always controlled, so their overall velocity becomes very uniform. And we can do also non-spherical particle inertial focusing is possible. So when we flow the disk versus cylinder, and then if we match a certain Reynolds number, they start tumbling and then flowing, and then they achieve the very uniform velocity. But when we consider what will be the characteristic dimension that determine their location across the channel distance, we found that as a particle tumble, they create some kind of artificial diameter. And then when we compare all this particle with the different shapes with respect of this artificial diameter, that we can predict that they behave like the sphere having the same diameter of that artificial tumbling diameter, except for one case when the particles start having symmetry breaking. So it's not symmetry anymore, they start behaving slightly differently. And then I'll show you other consideration that we're currently working on. So this will be another way that we might be able to control the surface area of the particle so that we can multiplex different type of assay using this artificially shaped particle without using color barcodes. And these are all the relatively rigid particle that's not capable of changing its shape in response to the shear force from the flow. But when the particle itself can be change its shape in response to the forces that apply to it. So these are the oil droplet flowing in water. So visibly might not be able to see how much is shear, but it's in response to the shear gradient, it can change its shape. But even though the droplet is very soft, we can still achieve inertial focusing that's confirmed by the uniform velocity of all these particles are flowing. So this looks like relatively large deviation, but if you look at how tight the velocity is distributed, it's really well controlled. It's the 17 centimeters per second for large number of the particles. But interesting thing about this particle being able to change the shape is that when the particle is exposed to shear, it changes shape. They find the location where they experience the least shear, so then to go to the center of the channel. So as a result, if you have wide range of the particle softnesses varied, then you can position their location across channel very differently. So when the particles are very soft, they tend to go to the center of the channel. When the particle is very rigid, they tend to go to stay near the wall. So as a result, inertial microfocusing can give you multiple knobs that you can control their location. So if the particles are slightly different in size, then larger particles tend to be a little closer to the center. But if the particle have identical sizes, but they have slightly different deformability, then the soft one tend to go to the center. But also if you increase the fluid inertia a lot further so that you can push everything near the wall so that you can uniformly control that. So that gives you multiple wall knobs that you can tune to position different particle uh, uh, populations. And whenever I talk about people that we're going to use biomedical instruments using the cells or other things flowing the meters per second in very tiny microfluidic device, the first thing that we're asked is, are their cells going to be survived through that? And then answer was yes. I kind of give you the preview first. And then we already demonstrated that their genomic markups is not really changing before and after the microfluidics. So C is control, F is in flow, and the viability doesn't change. And then we can collect the cells and then culture more than a week. So more interesting thing we found is there are differences more depending on the different flask that we're collecting versus control versus a flow condition. So with this preliminary results, hopefully I was able to convince you that inertial microfluidics can uniformly position particles if we need to, or differentially position particle depending on their size and softnesses. And also it's very biologically friendly. So we can use inertial microfluidics without concerning about biological sample damage. So I will show you some of the application that we're manipulating inertial microfluidics. So first one is how the uniformly positioned particle can be useful. So I'm sure you guys might know about flow cytometry. That's very, very powerful technique that you can do a lot of different assays. But in order to extrapolate all this information that biological sample carries, you have to position individual cells at the precise location where the laser can shine, and then you can collect the reflected light signal from the individual cells. So as a result, you have to use enormous amount of the sheath fluid to control the precise location of the individual cells. And then also you have to control different spacing. So because we have to use very clean solid solution, that's enormous amount of fluids, it's really hard to make it parallelized system so that you can use very small compact cytometry for a lot of different applications. So what we decide is, since we can control the particle positions without using a lot of sheath fluids, we decide to make a parallel microfluidic cytometry. And this is a movie that's showing individual dots are the blood cells that's collected from human blood. And then you see this kind of creating the traffic jam of the individual bloods. Everything is flowing uniform speeds, and then we can predict their location from the wall. So if you have a snapshot, you can, and then frame rates are matched, you can count or look at image of individual cells, how they are flowing. So these are also zoom in version of the movie, that when the cells are not inertially focused, they kind of flow random speed, and then they're overlapping each other, making almost impossible to detect which one is which. But when we do the inertially focused, that we can distinguish cell size more clearly, and everything is in focus image-wise. And then as a result, we could differentiate red blood cells versus white blood cells, and really high sensitivity and then specificity. And now I'm moving on to where we can use this differentially positioned particle could be used for. So there are a lot of diseases that change individual cells' mechanical property as disease progress. These are just some of the examples. So one of them is a cancer that when become more metastatic, they tend to be a lot softer so that they can migrate out of the initial tumor sites getting into the bloodstream. And also that when patients are infected with malaria, or they have sickle cell anemia, their red blood cell tend to be a little stiffer. And also the patients with the leukemia or other type of disease, infections or chemotherapy, they tend to be a lot stiffer or change their mechanical property. So if we can purify cells based on their mechanical property without using any other molecular label, they will give us a lot of benefit of purifying them and then understanding more afterwards. So we have conducted that using how the soft particle and large one tend to go to the center of the channel, while the small ones stay in the near the wall. So these are just scatter plot, kind of similar to the flow cytometry data. So the x-axis is a pretty much non-dimensionalized size, and this is location. And then if the cells are healthy or less malignant, they tend to follow the relatively linear trend that has increasing size versus location across the channel. And so one being a channel center. But when we chemically modify the cell to have a more soft phenotype, that they tend to be more center. So using that, we were able to make a device that just flowing them, and then all the soft cell tend to go to the center outlet, while the stiff or relatively small blood cell near the wall, so that we can guide them into different outlet with the high purity. So this video tend to be a little cloudy and then looks not pure, because we wanted to visualize how the cells are going to which outlet. So we put higher cell concentration more than the device is optimized for. So that's why it looks less purity, but we were able to demonstrate that we have high enrichment, high purity factor when we purify cancer cell from the blood. And also similar technique was used to purify different tissue when then digest tissue with a different lipid content. So we can purify more progenitor cells carrying less lipids versus more mature cells carrying a lot of lipids. So we could apply this for tissue engineering purposes. And then we're currently working on this to expand further or a different type of applications. So we're currently working on to investigate the effects of all this softness, because the cell have a lot of different characteristics. So how the viscosity contrast of the cell affects their behavior, or how the membrane stiffness affect their behavior. We're currently investing into more to gain the fundamental understanding of how cells will behave under this high inertial focusing shear flow. So another the thing that I promised is that we're going to purify the cell and then find the therapeutic options for the patients. For that particular application, we're interested in the liquid biopsy of the patients. It's because a lot of this bodily fluids that we can collect from the patient body can carry a lot of information, what's going on in this different individuals. And then particularly for cancer patients, the blood can carry a lot of information about the cancer progress. And then one of the things that we're particularly interested in are circulating tumor cell that migrates out of the primary tumor, getting into the bloodstream and circulating around. So we want to collect circulating tumors from the blood so that we can understand what kind of metastatic advancement is occurring in the patients without getting too much of biopsy or surgical interventions. Another reason why we're interested in a liquid biopsy is there is a lot of armies of the cells that we can use to target those advanced cancers. So one of the example are the cytotoxic T cells. So if we can process a large amount of blood really quickly to collect this information out and then train them so that we can feed back to the patients or find the right therapeutic option, it will benefit advancing personalized medicine. So for that, we were changing the geometry slightly of the inertial microfocusing device. So this is pretty much the same phenomena happen upstream, so where the straight channels are occurring, then two lift force are balanced and then make all the particle with the different sizes focus at their equilibrium positions. And then when the particles or cells are enter this uh, expansion region, then because we're flowing fluids really, really fast, there is going to be secondary flow is going to be formed. It's kind of like a swirl that you might have seen when you go to hiking, that when the rock is blocking the water, there's water turning around, and then you might see some of the small branches or trashes are trapped there and then circulating around. So we're creating a similar swirl in micro scale, single cell level, so that when the large cells are entering the swirl, they experience relatively large lift force, because this lift force pushing them towards the swirl is scaled with cube of the particle size, while the small one doesn't really get pushed further enough to be stably trapped there. So small one tend to go in and then flows out like this stream of the water collecting trash, while the water can get just freely flow out. So as a result, we can trap only the large cell from that heterogeneous solution, for example, blood of the patient sample, while this small freckles, those are all the healthy blood cells, comes in and goes out without getting trapped in this orbit. So using this technique also gives another benefit, that we can exchange the solution environment at the entire this small vortex region. The chemical environment can be switched within the 5 seconds without losing the cells that are trapped in the vortices. So when we change the chemical environment, the cells that are stably trapped stays, but cells that are not stably trapped can be all flushed out. So we can have very a pure population of cell remaining in this chamber. So these are videos showing that how this can be purified the cancer cell from the blood. This is a whole human blood, diluted human blood. And then when we trap the enough number of cancer cell, we switch to the clean buffer, then only the stably trapped the cancer cell remains, and then all the healthy blood cell goes out. And then since we're using only flow to trap the cells, so if you slow down the fluid, then this cancer cell goes out without any problem. So when initially we flow the blood and trap the particles or cells in the blood large enough so that it can get trapped, and then we switch the buffer and then only the cancer cell remains. And then when we slow down the flow speed, it flushes out. And this technology has been licensed and then commercialized by Vortex Biosciences. There where I was working briefly to work on a lot of cancer clinical trials. And so this device can purify a very small number of cancer cells from the large background of the healthy blood cells. And then those purified cancer cell represents pretty much similar morphology as the either primary or metastatic sites of the patient's tumor sites, or they also carry the similar or distinct mutational markup. So it kind of gives you ability to screen different tumor progressions as without doing a lot of painful biopsy or surgical testing. And also now we're currently interested in other population of cells so that we can train them to target cancer. So we're currently working on creating a cell size bandpass filter. This time, instead of trapping large cell, we're going to trap the small cells. So we were able to identify right geometry ratio so that we can flip the trapping behavior. So now we're trapping the smaller one while rejecting the large particle. So we can fine tune the fluid mechanics so that we can choose which particle population we trap. So now we're moving on how we're going to train these purified cells after we purify them from the blood. So we're using electroporation. This is a relatively old technique that a lot of biologists might be familiar with. So the concept is you apply relatively large electric field to the cell very short period of time, then you can tear the membrane of the cell very transiently so that you can inject molecules from the outside into the inside of cells. So we combine the cell trapping mechanism with the electroporation so that we know exactly what type of size distribution of the cell that we can trap, and then we know where the location of the cells are going to be trapped so that we can fine tune the electric field, and then we can exchange different solutions so that we can inject multiple molecule into the porated cells in sequence. So this is video, it's showing that how it works. So the cells are trapped, they're alive, we stain them blue up front, and we exchange the solution to the molecule that we want and apply electric field. Then it porates, and then molecule slowly goes into the cells so that we can it kind of indicates that the molecule are successfully getting into the cells. And then we can repeat this multiple times. So with the different molecules, so that this one also shows that it's slowly taking up different type of molecule in sequence. So as a result, initially we have only cells, but later the cell is going to take up different molecule inside them so that we can deliver a precise amount of molecule into that. So we can control the dose by changing either electrical field strength or the duration that we're going to incubate cell in the different chemical environment. And also we can fine tune the electric field because we can visualize a process so that we can choose right electric field for type of cells that we're dealing with. So without altering their viability, we can still deliver molecules into the cell very safely. And then now we parallelize the system so we can actually accommodate the patient sample. So using microelectrodes, making sure entire the channel will have uniform voltage across the individual electrodes. And then using that, we're able to deliver a different plasmid into the cell so that we can culture them afterwards. We can co-deliver different proteins, and also we can deliver microRNA or a silencing RNA so that we can selectively alter their phenotypes. And then now we're currently working on actually purifying cells from the blood so that we can do the electroporations onto cells that has been purified from the blood. And then we're showing that we can efficiently deliver the molecules without altering viability of the cells, without losing the purity of the cancer cell that we can purify from the blood. So with that, we were able to deliver a wide range of the molecules, and then we can choose different size distribution that we want to purify so that we can choose the cells that we want to get from the blood and then genetically modified or chemically modify them with a very controlled manner. So with that, hopefully I was able to convince you that inertial microfluidics could be very useful. So we can either position them very uniformly or differentially by using particles mechanical property or fluid property. And also using that, we can purify different population of the cells or particles from the very complex solution. And then we can alter their either genotype or phenotype by delivering different molecules in very controlled manner. So with that, I'd like to welcome any questions that you might have, but I also want to have a promotion of the microtask. This year, we're going to go on virtual for the first time, and then the abstract submission deadline is coming up soon. So hopefully you guys can join this year's microtask and enjoy virtual seminars. So with that, I'd like to answer any questions that you guys might have.
Awesome. Thank you so much. I'll clap on my side, but imagine.
Sorry, I went over a little high more. Sorry.
All right, great. So that was really a fun talk. I have some questions, but I think I'm going to go to the chat first to ask some of those. So our first question comes from Ambika Somasander. And her question is, what is the limit of size for soft particles beyond which they deform?
I mean, you mean deformation limit or size limit?
Maybe size limit where maybe you can't detect.
So, so we're doing it image analysis wise. So it's depending on if the particle is too small or a droplet is too small, even though they deform, we might not be able to detect it because of pixel resolution. So that's a camera limitation. But the smallest the particle we were able to focus was about 20% of the channel width. So if you're dealing with very small particle, you have to make channel very small. If you're dealing with large particle, you can make a channel a little larger, but it's non-dimensionalized, so that's why I say percentage. So you can scale up or down because Reynolds number is all non-dimensionalized numbers, so you can do that. But then the inertial focusing might not be able to deal with smaller than 200 nanometer or no, sorry, 20 nanometer, because then Brownian motion will kick in. So inertial focusing might not be able to applicable. And also technically, we might not be able to decimal of the channel that can sustain high pressure drop to flow meters per second speed in nanoscale channel. So that could be another technical limit. Hopefully I answered the question.
Great. Well, we'll check the chat.
Yeah.
I think that was pretty thorough. All right. So the next question comes from Brendan Hanrahan. And so the question is, can this approach provide a time savings to diagnosis for cancer metastasis?
So time saving, hopefully, eventually, because time saving means if we can do weekly assess the patients, if we can do it this way, right? So instead of biopsy, weekly might not be feasible. So eventually what we want to do is that we can monitor patient as they go through the treatment without going through a lot of painful procedure, but also purifying circulating tumor cell by itself cannot serve the diagnostic purposes. We have to do all this molecular analysis. So all those things has to be accompanied together, because even though we purify cancer cell within 30 minutes, if we can analyze cells for like weeks, they might not give you the time saving benefit of that. So that has to be all accompanied. But for just the purifying purposes, the instrument that I said that's commercialized can purify circulating tumor cell from 4 ml whole blood, undiluted whole blood, within 30 minutes.
Great. And then actually I had a question just about, so it looks like most of the demonstrations that you've done are using kind of standard rectangular cross-section panels. Do you think there would be any benefit to having really complex geometries as the channel cross-section, maybe like a star?
I'm sure it does, but then the analyzing them will be very difficult too. But my former colleague is actually working on the half dome shape or the triangular shape to see if you break further the symmetry of the channel cross section, how the inertial focusing changes. And then he has demonstrated that will actually change it so that it will give you a benefit of either focusing everything into one spot versus 3 different spots. So I'm all for any other opportunity if you want to help us building another geometry. But then the tricky part is we have to rely on image analysis, because there's not so much of the computational tool that we can predict much, because this is very complex fluid mechanics, with the particle being large and then its confinement and then the walls being really near and then particle itself changes shape. So we have to do the experimentally. So if we can image them with like very complex geometry without alteration, we might be able to see interesting phenomena.
Okay, interesting. Yeah. So that definitely makes sense in terms of the microscopy component of it being critical. No, that's great. So we're a little bit over time, so I'm going to end it there. Thank you so much, Professor Herr, for coming and giving this virtual seminar during these unusual times. Thank you so much. Next week, we're going to have Ambika Somasunder, who's going to be from Pennsylvania State University, who's going to be presenting on positive and negative chemotaxis of enzyme-coated liposomes. So thank you again, Dr. Herr, and thank you everyone for watching. And that is it for today.
Thank you,
Bye, everyone.
everybody.